thank you to CCF for the invitation to come speak today. My link to CCF and Giant is also that I was awarded a Next Gen Award, which has been tremendously helpful moving forward in my research as well. Today I'll be focusing primarily on monoclonal antibodies and leukemia, which Terry alluded to a little bit, but I think it's a really important topic as it represents a different form of immunotherapy that is becoming quite useful and relevant in the field of leukemia. So overall, this is a pediatric cancer incidence. Leukemia represents 25% of the cancer diagnosis in children. ALL in particular is the most common pediatric malignancy, affecting 75% of, which is about 75% of childhood leukemias. It has been the poster child for success and outcomes of therapy for pediatric cancers, and there's been a dramatic improvement in the 5-year overall survival. I think Terry showed a similar, or showed a slightly different slide. This just shows you the survival by study era. You can see here sort of dismal overall survival in the 1970s with rapid improvement to sort of where we are now. AML, which I'll also talk about just briefly in terms of antibody-based therapy, represents 20% of all childhood leukemias. But this overall survival is quite a bit less compared to ALL, with overall survival of 60 to 70%. Again, you have a similar improvement in survival by era, but again, very intensive therapy associated with high risk of infections. There's a 5 to 7% associated treatment-related mortality or patients dying from the therapy itself, much less in ALL therapy. And again, relapse is the greatest cause of treatment failure, with about 30 to 40% of patients relapsing with AML. Standard treatment course will consist of multi-agent chemotherapy. Outlined here is sort of the different stages of chemotherapy that patients go through for ALL. For AML, it's slightly different, but again, various stages of intensification. And I think it's worth mentioning just some of the side effects. So in terms of ALL therapy, patients will receive steroids. Again, acute side effects are going to be high blood pressure, high blood sugars, vincristine. They have a lot of sensory and peripheral neuropathy. Ankles may not work quite as well, constipation. Some of these peripheral neuropathies, because at the NIH we see a lot of patients who have had leukemia for 5, 10 years, and it's really significant neuropathy often at times. Asparaginase, you can have an immediate allergic reaction. You can have clotting factor abnormalities, stroke, pancreatitis. We have a patient now who has now a chronic history of recurrent pancreatitis from pegasparaginase. Methotrexate, again, these are all just the primary drugs used in ALL, high cure rates, but it comes with a lot of toxicity. Again, you can have a lot of neurotoxicity, headaches, nausea, vomiting. Doxorubicin can lead to late effects with cardiac toxicity, and then intrathecal chemotherapy, which is delivery of chemotherapy into the spinal fluid to both prevent and treat cerebrospinal disease, and can lead to significant neurocognitive effects as well. I didn't even mention on here not total body radiation, which can be used as part of transplant if it's needed, but just CNS radiation, which is also going to attribute to overall neurocognitive side effects over time. Late effects, I think, also worth mentioning. So anthracycline therapy, so that's the category of drugs that are going to be doxorubicin, daunorubicin. Patients, depending on the doses that they've received, sometimes even less than that, they can have problems with developing heart failure later on in life. Reproductive problems, infertility. Some of these patients will go on to getting a transplant that's been mentioned before. They may not be able to, may become infertile, may have secondary cancers as well. Radiation, again, total body irradiation was briefly mentioned. Whole host of endocrine abnormalities, cranial radiation I just alluded to. And again, just infertility. And again, some of these other chemotherapy agents, you can risk secondary cancers. Again, with the steroids, you see the patients can gain a lot of weight, have decreased bone mineral density. And these I both mentioned as well, but also as part of late effects, not only just acute. So the challenges in current leukemia-directed therapies that you have, there are a percentage of patients in ALL, it's about 1 to 2% of patients who are completely chemotherapy refractory, even at their initial therapy. Relapse disease, again, 30 to 40% in AML, slightly less than ALL, but those are the patients where it is really difficult to treat them. Cumulative toxicity, and so especially if you have either refractory or relapse disease, you keep sort of intensifying the therapy to put them into remission. The therapy can take a toll on all of the body's organ systems, making them less able to receive therapeutic options. And then, of course, there's the inability to receive all of the therapy. So if somebody receives peg-asparaginase early on, has an allergic reaction, you go to sort of another form of asparaginase to try to get all their therapy in, but there may be a possibility that they can't receive all the therapy, which does compromise their ability to have a long-term cure. So monoclonal antibodies. So these are, so we talked briefly about antibodies. I think everybody knows what antibodies are. But they are essentially agents that would recognize a cell surface protein or a cell surface marker that is more highly expressed on leukemia than on normal cells. They may serve to overcome chemotherapy resistance. They often have a very unique side effect profile. Again, chemotherapy, I sort of think of, you give chemotherapy, affects all of your cells pretty globally, both healthy and the abnormal cells. The idea behind monoclonal antibodies is that, again, you're giving very targeted therapy. But as a result, you also may have sort of unique side effects that emerge as a result of what you're targeting, or that emerge based on what the agent is combined with. So a lot of the monoclonal antibodies can either be given alone, just targeting a specific cell surface marker, or they're combined with some other, either chemotherapy or some toxin to enhance the potency of that agent. And then the other part of monoclonal antibodies is that they can be given alone as a single agent, or they could potentially be given as part of a combination chemotherapy. And the idea there would be to either enhance the efficacy of standard chemotherapy combinations, or potentially even replace one of the more toxic chemotherapies if you could. Shown here is sort of a general schema. This is mostly for ALL, but again, you can see here that these are sort of the different surface. Dr. Fry mentioned targeting CD19 and CD22 in terms of CAR therapies. Again, they're all different forms of current antibodies that are targeting. So for 19, I'll talk briefly about blinatumomab, as mentioned in some other therapies as well. For CD22, so that's one of the therapies that I've primarily worked on at the NIH, developing a phase 1 clinical trial, subsequently a phase 2 clinical trial with an agent called moxetumomab, which was conjugated to Pseudomonas exotoxin that I'll talk about briefly. CD20 is a little bit more broadly used, more primarily in lymphomas, where there's much more CD20 expression uniformly. In ALL, you can see it maybe 50% of the time, and so may or may not be as useful in ALL therapy. And then CD52 has sort of fallen out of favor, but you often will see it used in some transplant regimens. These antibodies can either be what we considered unconjugated, so just an antibody alone, or conjugated to either a chemotherapy agent, an immunotherapy agent, or what blinatumomab is, which I'll talk about towards the end, is a bispecific antibody where it really serves as a synapse to bring together your targeted therapy with an immune cell, so a T cell. I'm shown here are various mechanisms of actions of monoclonal antibody-based therapy. So you've got your basic structure of your monoclonal antibody with the FV, which is going to bind to the target, and different mechanisms of action. So you can have, it conjugated to a radioisotope. Again, not so much used in ALL, but in other therapies, you have yttrium binding. And so the idea is that you conjugate to the radioisotope, bringing radiation therapy basically directly to the cancer cell. You can combine with a constant or chemotherapy conjugate with, if that antibody is then internalized or the toxin is internalized, it goes into the cell to inhibit either protein synthesis or cause some type of dysfunction leading to cell death. It can have some growth inhibition. You can bring it together with another effector cell. So in the case of blinatumomab, we bring it together with the T cell. For those antibodies that are not conjugated to anything, they often will depend on the inherent immune system to play a role. So you might have the complement cascade setting off a series of events that would lead to cell death. And then also just antibody-dependent cell-mediated cytotoxicity. Again, you're relying on the inherent immune system in conjunction with the antibody-based therapy to lead to cell death. So this is a list of available therapies in ALL. The vast majority of these are still in clinical trial, but I think it's worth mentioning both rituximab, ofatumumab. Again, these are CD20, mostly really being used in lymphoma therapy, but has been shown to have some effect in improvement of survival, primarily in young adults with ALL. Again, they're both FDA approved. Blinatumomab, again, is a bispecific, recently FDA approved in December of 2014. a whole host of CD22 targeted therapies. Epertuzumab is not conjugated to anything. It's an antibody alone. Epertuzumab is now, with the SN38 is conjugate in clinical trials. Inotuzumab, I'll mention as well, is currently on target for FDA approval. It's sort of been designated as a breakthrough therapy. And moxetumomab is the agent that we've worked on in development at the NCI. So moxetumomab, I'll mention briefly. So this is an anti-CD22 immunotoxin. You can see the structure up at the top right there, and it binds to surface CD22. One of the things that we've done at the NCI is really establish expression of CD22 in ALL. It's actually not all that well established, and we have served as a primary sort of a central site for CD22 expression. And so oftentimes, especially in the development of this clinical trial, we would have a lot of sites saying, well, our patient's only 50% CD22 expressing, will this really work? And we would basically ask them to just send in a sample and show that, most of pediatric ALL really does have 100% CD22 expression. And that data actually was very informative to the future development of our CAR therapy targeting CD22, so that we knew we would be able to target the vast majority of patients with pediatric ALL. In the case of moxetumomab, so it binds surface CD22 and that whole immunotoxin, so again, it's a conjugate. Sorry, I don't know why I keep doing this. It's gone. Sorry, the top. I will not press the top again. And so basically what it happens is that whole immunotoxin is internalized. The toxin is then separated. It's released. And then essentially what it does is it inhibits protein synthesis, essentially leading to cell death. When we describe it to patients, we say it's basically starving the cell to death. So it doesn't work very quickly, but it can be quite effective. And you often would see patients, when we would treat them, that they would have an increase in their leukemia burden, but then after 3 doses, you would start to see it come down. This is a patient that we treated that I think was just showed sort of fairly impressive response. So you can see here, this is a PET scan. This is a patient, oftentimes our patients will show up with very unique presentation of their relapse. In this particular case, he had disease not so much in the bone, in the pelvic bones where we typically think of identifying ALL, but you can see here all of these black spots are areas where he had leukemia burden, all up and down the long arms, the legs, upper and lower extremities, all along the lymph nodes. And he had 2 cycles of chemotherapy of moxetumomab and went into a complete remission. And so based on the phase 1 studies, we had an overall complete remission rate of about 25%, and certainly enough of a favorable sort of a risk-benefit profile to warrant further studies. And it was an international phase 2 study. Unfortunately, in the phase 2 confirmatory study, the risk-benefit profile was less than favorable, and so it did close early. But what we learned from that was sort of, ways to either enhance the activity, and so it's now gone back to the lab where they're studying a novel formulation, thinking about a continuous infusion of this drug, how to further develop it. Because for those patients who it did benefit, the response was really quite tremendous. So looking at, basically novel formulations, maybe a new phase 1 study in the future. And sticking to the theme of targeting CD22, I think it is very important to mention inotuzumab because this is the furthest along in development. And this is one where, so going back to Moxie was really studied more in pediatric ALL. Definitely not, there is, I think, only one phase 1 in adults. Moxie, I should mention, is actually will be going for FDA approval in hairy cell leukemia, which is worth mentioning. Again, it's a leukemia that primarily affects older adults, where it's had about a 70 to 80% complete remission rate. So the agent will be moving forward, although maybe not in this formulation for pediatric ALL. Inotuzumab, like I was saying, is the furthest along. It's currently received breakthrough designation, and it's another anti-CD22. It's a drug conjugate. In this particular case, it's bound to a chemotherapy agent called calicheamycin. Too toxic to give, systemically, so, giving to somebody just alone, but in this conjugated form, again, similar concept, it goes into the cell, the chemotherapy then binds to DNA, leading to double breakage in the double-stranded DNA, leading to apoptosis or cell death. So just this past October, it got breakthrough therapy. So, phase 3 trials are really the gold standard in terms of, moving along to clinical trial development and getting drugs approved. And so this is an adult study, their Innovate trial, where they enrolled 326 patients, all of whom had relapsed refractory CD22 expressing ALL. They got, weekly doses, and it was a comparison of either inotuzumab given once a week for 3 weeks compared to one of 3 different standard chemotherapy regimens. And it was very clear that, the complete remission rate was very high, about 80% compared to 33% of those patients with relapsed refractory disease, of which 78% of those were completely negative. So MRD standing for minimal residual disease really means that absolutely no signs of leukemia at all. So again, a very effective therapy. But again, like I mentioned, some of these monoclonal conjugates, these antibodies, have very unique side effects. Again, this therapy is extremely well tolerated. You don't have the low cell counts, the thrombocytopenia associated with it, or the mucositis or infection risk. But in this particular case, probably related to the calicheamicin, their mechanism we don't quite understand is related to veno-occlusive disease. Going back to sort of thinking about curative therapies for ALL for patients who are either refractory or have multiply relapsed disease, we know that their disease will need consolidation and curative therapy with a transplant. Transplant is inherently associated with the risk of this complication called veno-occlusive disease, which at some points can be quite fatal. And so a lot of patients what we learned were, getting inotuzumab and developing veno-occlusive disease after transplant. I think the most recent number is about maybe 20%, and so a lot of what we need to do in the future is to learn how to best incorporate these therapies so that these therapies can serve as a bridge to transplant, leading to long-term cure. Inotuzumab in and of itself has not provided long-term cures, but it's very effective in getting patients into remission. Blinatumomab is sort of the most exciting antibody out there, and again, the difference here is that it's not monoclonal. It's really a bispecific T-cell engaging antibody. So again, I think of it as really serving as a synapse. It targets your CD3, which is your T-cell, and brings it into close proximity with your CD19, which is your leukemia cell, as well as your other normal B cells. And so it's really redirecting the T-cells to sort of say, come along, here's a leukemia, do what you need to do, and really sort of helping them. And I think one of the issues with blinatumomab has been a lot of our patients have really low lymphocytes. We aren't able to, even collect their lymphocytes enough for CAR T-cell therapy, but at least with blinatumomab, there has been some thought in the preclinical setting that it may even help to induce T-cell expression, which may help in the overall efficacy, because it does rely upon having somewhat of an intact, partially intact host immune system. It's a very interesting concept in terms of its infusion. And this is a model that moxetumomab may eventually may follow. But it's given as a continuous infusion over 28 days. So, a model that we have never really done before. And you have these patients with these little pumps walking around, and they're getting this continuous infusion. And all of this is really based on, the pharmacodynamics, how the drug works. It had initially, in its initial development, been given as bolus infusions, and the toxicity wasn't amenable to that infusion. So based on the overall response rate, it did receive FDA approval in December of 2014. It is currently in several phase 3 trials, and the main side effect, again, it would be similar to the CAR T-cell, is cytokine storm. And so a lot of it is, again, as this drug becomes available, is having people who are familiar with giving this drug, understanding the toxicity profile, knowing how to treat patients who are developing the toxicity profile so that you can overcome the toxicity and still achieve maximal benefit. This is the structure and function. So again, I mentioned it sort of serves as a synapse. You have your blinatumomab, which is the anti-CD3, anti-CD19, leads to the T-cell activation, and again, you have your target cell, which has a CD19 on it. And you're redirecting sort of the cell lysis. Overall response, so the initial studies really showed this really nice response rate of 70%, which I think was, again, when you have something that was as comparable to CAR T-cell therapy, and again, in phase 1 settings, to have something like this was really phenomenal. In the confirmatory study, again, I think when you treat more patients, when you expand that, you almost always see this, where it's about 43%. Again, this here is really commenting on adult patients. In pediatric patients, the phase 1 studies and subsequent phase 2 also had about a 30 to 40% complete remission rate. But again, really nice response rate compared to what other phase 1 studies had shown prior to that. And then gemtuzumab, I think it's also worth mentioning. And so this is shifting gears a little bit into AML targeted therapy. So this is an anti-CD33, and CD33 is an antigen found on AML. It is conjugated to calicheamycin, and so if you remember, I mentioned calicheamycin is as present as a conjugate in inotuzumab. And it has been a really nice option in AML. It actually received, it was one of the first antibody-based conjugated antibody therapies that received FDA approval. It was under accelerated approval in May of 2000. So again, quite a few years back, because the response rate was so impressive, especially in a disease such as AML, where the overall response rate was so poor. But it actually ended up being voluntarily removed by Pfizer in June of 2010 due to safety concerns. And again, VOD was this major issue, and so there was a children's oncology group trial for AML, specifically looking at gemtuzumab as part of one of the induction therapies. And those patients who went on to transplant, there was an increased risk of fatal VOD. And so there's some thoughts about how to look at this drug, about whether it's going to come back into the market, how to gain more access to it, because it was, really phenomenal therapy, but you again needed to figure out how to manage the toxicity in the long term. So in summary, antibody therapy may provide a synergistic mechanism to improve outcomes, may have a different, potentially less toxic side effect profile, certainly a different side effect profile. It can enable treatment of chemotherapy refractory disease. And again, at the NIH, we are the ones seeing a lot of these patients who have either had multiply relapsed disease or chemotherapy refractory, and provides a targeted approach. So aside from blinatumomab, all of the other antibody conjugates that I mentioned, I just focus on sort of the ones that are the most developed overall. They really don't depend on having an innate immune system. And so they come in with their own toxin or their own immunotoxin and can function without needing to have an intact immune system, because a lot of the patients who come to us, they've come pretty beat up after receiving a lot of chemotherapy already. I think future directions, what we really need to do in terms of antibody therapy is to really, to understand how to optimize it. Right now, the data that I've presented really has been data presented when it's given as a single agent. And you sort of want to think, well, if it's this effective as a single agent, how much more effective could it be when given as combination chemotherapy? And some of those studies are just really starting to be done. And again, a lot of it has to go back to how, there is drug development, getting FDA approval. You start with a single drug and then you combine it. But I think a lot more needs to be understood. And I think ultimately what I would envision as a future for antibody therapy is that, if you go back to that slide where I mentioned the late effects and the, cumulative toxicity, if you could, replace one of those agents with one of these, one of the antibodies, you might be able to really change some of the long-term side effects for patients with leukemia. Understand the toxicity profile. So again, there are very unique toxicities. Cytokine storm with blinatumomab. I didn't mention moxetumomab, but we saw very unique kidney toxicity related to, low platelets and, breakdown of red blood cells, which again was one of the dose-limiting toxicities for the moxetumomab. The veno-occlusive disease, we don't really understand, but I think understanding the mechanism of toxicity is what is going to help us to overcome that profile. I think we need to think about what are the best uses also of antibody-based therapy. So again, thinking about, transplant as being the curative to consolidate leukemia-based therapy, we really want patients to have absolutely no evidence of disease. And so in that setting, some of these antibodies have been really effective in getting rid of, even the lowest level of disease, and that may serve a really great role. And I think we also need to understand resistance mechanisms. Terry mentioned briefly about blinatumomab and that leading to some degree of CD19 negativity. We are just, we have a lot of patients now who are receiving inotuzumab who may potentially go into CD22. They retain CD22, but we are starting to see maybe some differences in the pre and the post-inotuzumab therapy as it relates to how much CD22 they have on it. We don't know if these patients would become resistant over time. And so I think that's a way we need to understand. Oops, that's my last slide. And I turn this off again. So I'll take questions.
rate is interesting. I mean, you mentioned the difference in response rates in the subsequent follow-up trials compared to the initial trial, and it does seem to me that that the response rate in pediatric ALL with blinatumomab seems to be lower than adult. I mean, is that? Does anybody have any thoughts on why that might be? I mean, are they more heavily pretreated? I mean, it's.
I think that's a leading thought about why it is. I think some of the adults. So by the time adults are relapse refractory, they are much earlier on in their therapy, and so adults just don't do as well with ALL therapy. And so I think the children who come to blinatumomab, they're much more beat up than the adults are, and so I think some of the primary studies done, that had much less salvage attempts than the children. So I think there is a thought that has to do with the inherent immune system.